Hope you can see it. So I hope you can see it. Yes, and thank you very much for the nice introduction. And it was much too much about me and Steffi. And I heard a lot of big data today. And now I will present you something, only 3 ratios, hydrogen, carbon, and nitrogen. And I will show you something we can do with it, but something also where we can must be carefully. So I will start with some examples and first a little introduction. So the underlying principle in chemistry is that bonds between heavier isotopes are stronger. So if you have a chemical reaction, in the beginning, you have always a discrimination of the more heavy isotopes. And I will give you an explanation for this. But in the beginning, I will show you that these isotope ratios are very difficult to determine. So we cannot measure isotope ratios of carbon 30 to carbon 12. The ratio is very, very tricky to determine. Has been done maybe 5 times in ever. For example, here I have a value where I show you, this is of the reference standard we are using. Because of this problem that the absolute values are difficult to determine, we present relative values and we present them on a delta scales. And that means we measure a sample relative and parallel to a standard, and then we receive the delta, in this case 13, the C value. And there's one thing which we have to bear in mind in that context, that the standard we are using is an international standard. The Vienna PD belemnite is very, very heavy in carbon. So that means you have a very, very heavy standard. And if you subtract this standard from the sample, and if the ratio is very high in the standard, you get a negative value. And this is the reason why all, more or less all samples we are seeing now are negative, with negative delta values. And the more negative, the less certain carbon you have in a sample. So I will repeat that again. Now let's first start a little bit with chemistry. If you look at the chemical reaction, you have an adduct and you transfer it to a product. And in a closed system approach, so that no carbon is lost, when everything is consumed, the product will have the same isotope value as the adduct. You can say this. So this is quite simple. And at the end, you do not have adduct anymore. And in the beginning, you do not have product. So you have in the beginning, if no carbon is lost, the same value as you have in the end in the product. But if you look at the progression, if a compound is reacting, as I told you, there are heavier bonds between stronger bonds between heavier isotopes. That means the lighter isotope is reacting first. That means when you have not full the reaction, but in part, that the adduct is steadily getting heavier. So from the beginning, it's steadily getting heavier until it is fully consumed. On the other hand, the product in the beginning is getting very, very light, because at that point, very light carbon is converted. But then afterwards, also the more heavy carbon has to be consumed. So that means that the product is getting also steadily heavier until in the end, all of the adduct is consumed, there's nothing left, and you have the value of the beginning. So this is the underlying idea, and should give you an idea what you can do with that. For example, if you have here a ratio of 25% adduct and 27% product, you can calculate with 0.25 times 22 means 5.5 per mil out of 28. That means the remaining share of the 28 must be in the product. That would be 22.5 per mil. And then if you calculate up, then the product at this stage must have delta 33 value of minus 30 per mil. And so you can calculate all that ratio, and then this can be considered in biochemical reactions and also in chemical reactions, and you can take advantage of it. And this could also be a step forward later in food authentication, where you could use such data, but this is maybe too early to consider all that. I can give you an example. If you have a chemical reaction, for example, the bromination of a diphenyl ether, then that means that the more heavy the compound is, that means the more bromo you are introducing, the lighter the compound is getting. But on the other hand, if a compound is degraded, it's the other way around. So you can also use this data to investigate if a compound is a metabolite or is it a naturally produced compound if you look to environmental sciences. So you have a lot of opportunities if you look at the isotopes, and all what I showed you now is related to the point that you have more stable bonds between heavier isotopes. And again, the more negative the value, the less 13C. Also, if you look at CO2 assimilation or sequestration, this is very important because again, the CO2 is the 13th CO2 is discriminated. So more 12C CO2 is fixed than 13C. And if you look at it in the atmosphere, you have a value currently of -8 per mil. If you look a little bit back, let's say 15 years ago, the value was -7 per mil. And this is the consequence of the human impact of the climate, because we are releasing technogenous CO2, which is lighter in carbon, and that makes on the long term that the atmospheric CO2 is getting also lighter in carbon. So at the moment we have -8 per mil, but it will increase if we continue with that. So also this technique is very influential to predict and to confirm the human impact on climate. Now, as I told you, the stronger bonds between heavier isotopes lead to discrimination of the 13th3. If you have the atmospheric CO2, I showed in the last transparency of -8 per mil, in C3 plants, you have values of between -20 and -30 per mil. So you see that discrimination in 13th in the consumption of CO2. And if you look at fossil fuels like biomethane, you can say you have an average value of -45 per mil. I will come back on these values later. Now I will give you the first example when we of a study we confirmed. We looked at red bell pepper from Germany, so grown in Germany, and the basic point is bell pepper is a C3 plant. So the value should be between -20 and -30 per mil. And this is the lower limit of C3 plant, which you see here. But if you look at the values we measured in the bell pepper, the values were much lighter in carbon. And so that does not fit at all with these 3 plants. So the question was, what is happening here? And to give you another example, most what I will show you today are bulk values. That means we measure the whole sample. But of course, we can also determine by GC IMS coupling the individual compounds. Nowadays, it's also possible, of course, with LC IMS. And then you can measure the individual compounds and the fatty acids in the bell pepper were the lightest measured in any food sample. So that means there was something very, very strange. These 3 plants which do not behave like C3 plants. So what is the story behind? Bell peppers in Germany are raised in greenhouses. And what is particular with bell houses and greenhouses? So there are several points to be considered. For the optimum growth, you need a heating and addition of CO2. And then this heating and also the addition of CO2 can be obtained by combustion of biomethane. And the biomethane is much more depleted in the compared to atmospheric CO2. So let's show you a calculation. If you have the source, the atmospheric CO2, the expected value in a C3 plant will be about -27 per mil. So you have a difference, discrimination of 13C by 19 per mil. Now, what did we find in the bell pepper? We found a value of -46 per mil. If you continue like that, what is the source? What we can see from the difference is that we expect that the difference will be the same. So the source should be minus 29 per mil to generate the value we measured in the bell pepper. And so the shift in the source causes the shift in the plant. So some data about greenhouse practice. If you double the atmospheric CO2 concentration, you have the highest yield of plant growth. So that means also at the moment, for example, for looking at the climate changes, at the moment we are increasing the level in the atmosphere of CO2, and this leads to a higher growing rate of plants in most areas. But it is predicted that about in 2030, it will be the other way around. And after that, the plant growth will deplete, so we will get problems. But anyway, doubling the CO2 concentration in the greenhouse will give you the best yield. And also the grow rate will be best for bell pepper at 28 degrees. And all this can be achieved by combustion of biogas. And that CO2 generated may be also released into the greenhouse. And there's a last point I want to highlight, that greenhouses may be open from time to time for better bell pepper growth. So you have 3 points: increasing CO2 concentration, heating, and opening, closing. Now let's go deeper in that calculation. I already showed you, if you look at the atmospheric CO2, which is of course also present in the greenhouse, we should have a value in the plant material of 27, minus 27 per mil. If you look at the prediction, if you take biomethane, the biomethane has a value of minus 45 per mil. If you look at the discrimination, the plant material should have 64 per mil, minus 64 per mil. Now, if you mix both of them 50-50, as I told you, which is the optimum ratio for growth, you will have in the plant material the value we observed in the bell pepper. So this is the explanation why these values in the bell pepper are so light. So by means of that, we can show that a plant is grown in the greenhouse and not in the field, which could also be very interesting, of course, as a new point of food authentication, because also that can be something interesting. Is it from the greenhouse or is it from the field? Now let's look a little bit more on that. If you look at the variations, we have seasonal variations. So we have here the summer, where we have a wide range. We have in the autumn, where it was comparably rich in intensity, and in spring, where it was light in carbon. So how we can explain that? And this is the third point. So we have a dependency on the season, and we can explain that by what I told you at the beginning, when the greenhouse is opened or closed. If a greenhouse is closed for 24 hours, and we have the same principle as I showed you at the beginning, then 50% will be atmospheric CO2 and 50% will be added CO2. And then you will get the value I explained to you on the last transparencies of minus 45. But when you open the greenhouse in autumn, for example, then it is opened only for 8 hours and it's 16 hours closed. This will, of course, increase the share of the atmospheric CO2 because when it's open, it's more from the atmosphere. And then the overall value will be lighter. So in autumn, you can expect lighter delta 33 values than in spring. And in summer, you have the whole range because also the greenhouse can be opened more frequently. So after all, we had a good explanation for that, for these very unusual 33 values in greenhouse. And then we compared it with this other samples. And if you look at Spain, the Spanish samples, they were all in the typical range of C3 plants. So there was no added CO2. And this confirms the field cultivation. If you then look at the Netherlands, in the Netherlands, we had a scenario which was similar to the German ones. So depleted in 33 So below the values you usually have for the 3 plants, that means grown in the greenhouse, that means also added to CO2, similarly to Germany, but obviously slightly different and overlapping conditions. And in that case, if you look at more delta, if you also consider the nitrogen isotopes, we could fully separate the 3 samples irrespective of the season. And so we have to talk a little bit now about the nitrogen isotopes. First of all, the nitrogen isotopes are used for trophic positioning. So you can find out on which level is an animal consuming. And by using the delta 15N value, you can see that the squid, for example, compared to the sardine is enriched in 15N. And this is because the delta 15 value increases by about 3 to 5 per mil per trophic level. So that means that the squid is feeding on a higher trophic level than the sardine. This is also a point which can be found out by the stable nitrogen isotopes. And the second point is the fertilizers. Fertilizers are different. So the artificial ones are rather low in 15N, while the organic one is in levels at +7 and even higher. So in this case, we are on the plus scale, but it can also go down to the minus scale. Also, again, the higher the value, in this case, 40 means more 15N. And if you look at this, we can say that in the Netherlands, the samples were raised with artificial fertilizer, and in Germany and Spain with organic fertilizer, or not by the addition of special fertilizer. So in that case, we could separate all 3 types of sample irrespective of the season. You can see that here, and I will come back on that because it's not always as easy as you will see here, that you can see that with the organic fertilizer. Now we come to the third point, which I wanted to explain you and discuss with you, is about the delta 2 value, so the hydrogen isotopes. And here it's more the physical isotope fractionation, which is important on the first glance, and that has to do with the transport of the water from the ocean and to the rain. So if the water is evaporated from the ocean, you have a vapor which is light on carbon, and then it is raining. And the more you get inside, the lighter the vapor will be. And so the more distant you are from the coast on the first approximation, the lighter the water is in hydrogen isotopes. And it also depends, of course, of the landscape structure, as I will show you now. So the rain is also, the more remote the rain is from the coast, on the first approximation, it's getting lighter in hydrogen. Now let's look what is happening in a plant if you do that. There's no isotope fractionation from water which comes from soil into the roots. So by entering the water into, for example, a tree, there is no discrimination of hydrogen isotopes. But within the plant, if you are forming organic compounds from the water, where the water is involved, there's a depletion of about 100 per mil plus a value which is dependent on the special situation and also on the plant. But at least there's a difference of about 100 per mil. So the delta 2 value in plants is the water value plus 100 plus somewhat. And this somewhat has to be determined. So what we did, we measured in the food profiling project walnuts, and we measured them, of course, also on nitrogen, I will show you later, and of course on hydrogen. This is the topic now. So now we had 6 points in Germany where we had authentic samples from, and on the first glance you would say, here's the coast in Germany, that the farther you get to the south, the lighter the compound should be in hydrogen. But that was not the case. So there must be a second point inside, not only the distance to the coast, and this is something we explored in that project. So there's no strict north-south correlation, which we expected, but what? So we had to learn something about the situation at the different sites. But unfortunately, we had no water data from the sites. So we should have soil or water from the site to correlate our values, but that was not possible. So what did we do? We looked at the global network of isotope investigation. This is a worldwide database which is collecting isotope data. You can form isoscapes of the whole world. And now we looked at, you see the 6 pointed with the walnuts, the brown ones, and then we looked to the published data in the database, which was closest to the site where we were analyzing. Then we were connecting the individual points, and then we made a straight line. So you get a fixed profile with altitude. So it's not only that you look at the distance from the coast, but it's also the altitude which is considered. And you can see here the impact also on the delta 2H value. And so you have a north-south gradient, but in addition, you have an impact of the landscape altitude. So the altitude and the distance from the coast have to be considered both, that you can work with hydrogen isotopes. And in the first 4 points, we had an excellent match between the discrimination between the water at the reference station and the walnuts of 110 per mil plus minus 3 per mil. So this is a very, very good match, so that we can say that for these 4 points, the isotope data we measured in the walnuts representative. But if you continue to the other points, you see there's a distance between the reference data of the water and the walnuts. And this made that the value did not really match. So the better data you have for the soil water, the better you can predict the value of hydrogen. And this is the tricky point. If you do not have that point, the calculations are very, very difficult to make, or other way around, it's very, very difficult to interpret the data. And you can also see the 6 point. Also here you have a mountain chain between the 2 measure points, and that makes a difference. But if you can eliminate that, you have always distance of 110 per mil, and then it is more or less the value of the water which gives the value in the sample. So then if you consider that, you will be able to distinguish the individual values from each other. And another point is very important, because as I told you, we are talking about rain, about precipitation. And here you see in that study, we had walnuts from 2 subsequent years. And what you can see here, that there's a huge difference between the 2 years. And so if you just look at the data, it is meaningless to compare this data. And you might think, okay, what can you do with the delta 2H values if you have such a variation from year to year? But you can consider that, because if you look at the rain data from the German Weather Service, you see that in 2017, it was much more raining. And in the case, if you have fewer rain, the water is transported farther from the coast, and then it is switched in 2H at the same place. So this is very important that you also consider these points in order to interpret the hydrogen isotopes. And of course, the same is true for oxygen isotopes if you want to look at the water. So it's not, you cannot only use the data directly, but you must study what are the environmental conditions because this, as I told you, is a physical fractionation, and therefore you need to know what is inside. And but if you go further, you can see differences between France and Germany in the walnuts, so all that can be considered. This has something to do that France has a longer coastline, and therefore you have a stronger impact, and therefore the values are rich in deuterium than the German ones, which are more remote. Short interruption, but just only the results. With that method, we could also separate Chinese travels from European travels. There's a very, very good separation between both of them, absolutely no overlap in the years of 2017 and 2019. But of course, what you have to consider from the last story I showed you, that it could be different in 2020 or 2021 because of the impact of the amount of rainwater and the transportation. Now, in the walnuts, we were also looking for organic walnuts. And as you can see here, this is German samples, organic walnuts and conventional. And you see a very, very good separation of organic and conventional. But if you look at the values in the organic one, -8 per mil has nothing to do with the organic fertilizer, which I described at +7 at least. So in that case, you have organic walnuts, and the value, the delta 15N value was -0.8, completely different to the organic nitrogen. So what is the origin or the reason for that? This can also be very good explained, because organic farmers, they have to check that enough nitrogen can be fixed. Therefore, they add crops in symbiotic relationship with N-fixing bacteria. And in that case, where you have the walnuts, it was alfalfa. And the alfalfa is on the same field as the tree, and this is the actual nitrogen source. And the alfalfa has a delta 15N value of -1, and this matches perfectly with the value you have in the trees. So, and that means that it's not so simple to look at organic fertilizers anymore. As you could see, it worked in the first study with the bell pepper, but if you look, for example, here at the walnuts, it didn't work at all, but still you could see the difference. So it's very, very important that you look at the special conditions at the field to interpret the data. Also, we had some values we cannot explain at the moment, but if you look at the travels and look at the delta 15N values, we had typical values of 6 travel species, which were all in the normal range of organic or natural soil. But there was something very, very special with the T. magnatum. That was much richer in 15N than the other species. And these samples were from, I think, 5 different countries. So it has nothing to do with the country. This is something which is very, very specific with that type of truffle. And you do not find that with any other truffle. We have no explanation. There must be something very, very specific with the host that you have such a high 15N content in this truffle species, while the others are not distinguished. And the interesting thing is that the magnatum is one of the 2 very expensive and very special truffles. So this is a very, very good marker where you can check it. But of course, at this point, we have to find out what is the reason for that. Finally, just 2 examples. If you want to distinguish organic and conventional, in that case, it is milk or cheese. It is, of course, in many examples, it's not so easy, because you have some samples always in between, which look more than the other one. And of course, if you have conventional cows, look at, for example, Highland cows. These are also feeding on grass, but they are not organic cows, so they will have the same values. So in such a case, only very simple examples can be distinguished from each other by means of the stable isotopes. But if you combine it with other data, like for example, such ratio as you can see here, you can find out, for example, what did the cow consume. So for example, we can, depending on these two factors, we can say that the feed was rich in grass or rich in hay or rich in grass silage. And to go back, grass silage is something considered not very good for organic farming. But if you have the delta value, if you are feeding grass silage, it will look like an organic sample. So the more data you have, the more detailed things you can say about the sample. And also back to the thing I started at the beginning, you can also do that in biochemistry. For example, if you have an unripe plant and you have a phytol is a compound of the chlorophyll, you all know, and the phytol is transferred later in tocopherol, but also in phytol. And if you have an unripe bell pepper, or in that case, chili pepper, at the beginning, everything is in the chlorophyll, and it must have the delta value. And then if it's reacting, it's getting more heavy in carbon if a share of it is reacting, and the products will be lighter. And at the end of the reaction, when everything is transferred, you will have the value which you had in the beginning, starting from the chlorophyll. And this would be, again, the closed system, which means that no carbon is lost, so that all is transferred into the respective products. And you can see the difference between unripe, semi-ripe, and ripe plants. And you can take advantage also of this. And these are points which can be the future in isotope data, that you combine these biochemical factors, which the values you have in the beginning. So a short summary of what I wanted to show you is that the stable isotope data successfully applied to different scientific questions. For example, greenhouse and field I could show you. And what I would show you is that stable isotope ratios are more than just values. You can take advantage of it. And frequently these tell a story. And the point is to unravel the story behind it. So why is something, for example, not as expected? What is the story behind? Still, it's only 3 values on a quite narrow scale. So in combination with other isotope data, or especially with other markers, they will be much more useful in food authentication, also in chemistry, biochemistry, etc. So the more data you have, and this is maybe the link to the beginning, what Markus Fischer said, that you can get big data, but it is very wise, in my opinion, to incorporate also this data. Finally, I want to thank Steffi Krauss, who did most of the study in that. I want to highlight the food profiling project and also the support in that project by the BLE. And of course, I want to thank all the co-workers who were contributing to that. And I thank you for your attention.